In this lesson, we are going to look at membrane structure. The essential idea that IB wants us to take away from this is that the structure of biological membranes makes them fluid and dynamic. So today we're going to look at the structure. What are the pieces and parts of biological membranes? Remember that biological membranes are not just the cell membrane, but also the membranes that make up organelles and eukaryotic cells, like the endoplasmic reticulum and the nuclear envelope and chloroplasts and mitochondria, all those organelles that have membranes. This structure is common to all of those things. And the structure makes them fluid and dynamic. Fluid, they can flow around. They're not stuck in a single spot. Dynamic, they can change. Good. So we're going to start with a couple videos. This first one is going to look at the fluidity of the plasma membrane of a nerve cell.
To demonstrate the remarkable fluidity of the lipid bilayer, a piece of the plasma membrane of this neuronal cell is picked out with laser tweezers. The membrane is pulled out into a tube, and the tube is moved around.
So clearly, the membrane is fluid. In this video, we're going to look more at the fluidity of membranes, but perhaps it seems to be that not all membranes are equally fluid. So what's going to happen in this video? In the lab, a protein that we're able to manufacture called GFP, or green fluorescent protein, GFP, is bound to proteins that are already stuck in the membrane of this cell. GFP, as you can probably guess from the name green fluorescent protein, glows under special microscopes. And so the scientist is going to kind of kill off some of the glow in the darkness of some of these proteins. And then we wait to see what happens as the membrane is moving around.
The lateral mobility of membrane proteins can be measured in living cells by FRAP, which stands for fluorescence recovery after photobleaching. For this purpose, membrane proteins are often expressed as fusion proteins with the green fluorescent protein GFP and observed with a fluorescence microscope. A selected area of the cell is then bleached with a strong computer-controlled beam of laser light. Those membrane proteins that are not anchored and therefore can diffuse in the plane of the membrane quickly exchange places with their neighbors and fill back in the bleached area. From the rate of this fluorescence recovery, the diffusion constant of the protein can be calculated. Here, GFP is fused to a membrane protein that lies in the membrane network of the endoplasmic reticulum. After bleaching, we observe quick recovery of the fluorescence, showing that the protein is very mobile in the plane of the membrane. The same experiment can be repeated using a protein that is firmly anchored and not free to diffuse. Here, we observe GFP fused to a protein of the inner nuclear membrane that binds tightly to the meshwork of the nuclear lamina. After photobleaching, no fluorescence recovery can be seen over the same time frame.
And so even though the membranes are fluid, not all membranes are equally fluid. The endoplasmic reticulum moved a lot. The proteins here in the nuclear membrane, really not moving at all. Good. So let's talk about the structures, the pieces and parts of the membrane that make them fluid and dynamic. The probably most basic component of the cell membrane is the phospholipid molecule. And this is what we have on our screen now. So we have here on the left the more atomic structure, whereas here, and it's still atomic structure, but we call this space fill. So you have a little bit more of an idea three-dimensionally what this molecule might look like. So it is called a phospholipid because there is here at the head a phosphate group. This is what makes it phospho. And then these two tails are fatty acid chains These are lipids, hence phospholipid. What's kind of amazing about these molecules being phospholipids is that phosphate groups are hydrophilic. They dissolve well in water. They're attracted to water because they're polar. There are some charges here that make them amenable to hanging out with water molecules. Whereas these fatty acid chains are hydrophobic, they repel water. And so we would call this molecule amphipathic, amphi because it is a both feeling kind of molecule. It feels both a like and a dislike for water. What that means is that it's got some pretty amazing properties related to polarity. So quick review of chemistry here. So nonpolar versus polar versus charged particles. So nonpolar particles have the same kind of charge all the way around the molecule. So if we're looking at this molecule of carbon dioxide, these oxygens have lots of electrons hanging out on the outside of them. And so the whole outside of this guy is negative, which means that if another CO2 molecule were to come along, that negative would repel. They're not going to stick together like little magnets. Whereas over here, if we're looking at water, the oxygen side of water has a partial negative charge. That's what that delta minus means. Partial negative charge, whereas the hydrogen side of the molecules have a partial positive charge. And so if another water molecule comes along, or this ammonia molecule, the positive end of hydrogen, the negative end of this nitrogen in ammonia, stick together like little magnets. This makes it polar. So polar molecules have a magnetic, we call it electrostatic, force of attraction that helps them stick together a little bit, whereas nonpolar things do not have that stickiness. Because the lipid part of our membrane is very nonpolar, things that are polar, these guys are actually repelled by this nonpolar middle of the membrane. Oxygen gas and carbon dioxide gas can diffuse right through. They're nonpolar. They're not going to be repelled by this middle. Small things that are polar can get through. So water, ethanol, which is alcohol, they can diffuse through the membrane, but a little more slowly than can the oxygen and the carbon dioxide. Big polar things like sugar totally get repelled. They are bounced off. And so sugar cannot just diffuse through the membrane. It needs a special kind of transport protein to help it get through. And then charged molecules, ions that have charges, pluses and minuses, are also going to be repelled by that nonpolar lipid bilayer. This allows the membrane to selectively allow some things to pass and some things can't. So our understanding of what membranes look like, the structure of the membrane, has changed over time, as all of science has changed over time, generally because of advances in technology. So back in the early 1900s, prior to the 1900s, it was understood that the membranes had to be some kind of lipid, since polar molecules were repelled, couldn't just pass through. So maybe it was just one layer of lipids, and then in the 1920s, it was discovered, made sense that there had to be just a lipid bilayer. There's got to be some kind of bilayer going on. In the 1930s, a pair of scientists, Davison and Danieli, noticed that there were proteins also incorporated into the phospholipid bilayer. And so they made a sandwich kind of model of the membrane. And it made sense because if we look at the pictures that they were able to take of membranes, there's clearly a layer of protein here and a layer of protein here. This sandwich model made sense. But then technology advanced, and we were able to do a thing called freeze fracturing. And what we were able to do was to literally freeze the plasma membrane and then fracture, split it right down the middle between the bilayer. So we were able to separate those two layers. And had the sandwich model been accurate, the inside of the phospholipid bilayer between the bilayer should have been smooth. It should have been if the sandwich model were accurate. We should have seen just lipids in here. It would have been quite smooth. But instead, their images were full of these divots, which led them to believe that the proteins were not just on the outside of the lipids, but instead were embedded within it. So this freeze fracturing technique allowed a different pair of scientists, Singer and Nicholson, to develop instead the fluid mosaic model of membranes. And we certainly have been able to better understand our proteins. We understand the structure of them better, and so we continue to develop this model. Science doesn't ever stop. We don't prove a thing and then be done with it. We're always trying to understand better and better. So again, we went from the Davison and Danieli model, the sandwich model, to a more accurate, given our understanding right now, Singer and Nicholson fluid mosaic model. Which brings us to this idea of the nature of science. It doesn't stop. We continue to develop technology which allows us to make new observations, which means that we can understand phenomena better. And so some of the evidence that we were able to use to falsify Davison and Danieli's sandwich model, that freeze fracturing of the membrane. We figured out that membrane proteins were globular, not fibrous. We'll talk a lot about proteins in our next unit, but this is globular protein. It's kind of globby, whereas this long stringy guy is fibrous, globular protein, fibrous protein. And then we were also able to do that video that we watched at the beginning of this lesson, that GFP green fluorescent protein tagging, to visualize how those proteins were able to float around. So these membrane proteins, these proteins that are embedded within the membrane, let's talk about some of their jobs. The functions of membrane proteins. Sometimes they stick two cells together. So a protein in one cell, a protein in another cell will hold on to each other to hold two cells together. Some proteins will act as enzymes. They might be involved in transporting things across the membrane, especially those polar and charged particles that can't just pass through the phospholipid bilayer. We use these proteins in cell-to-cell recognition quite a bit. So how our immune system knows what cells don't belong in our bodies and need to be attacked is with these membrane proteins. Sometimes the proteins are going to attach or anchor other stuff inside or outside the cell, so this anchorage. And then there's also this idea of signal transduction. So we might have a hormone come and bind to a membrane protein, and that protein tells something else on the inside of the cell to do things. So the hormone doesn't actually get into the cell, it just binds to the protein, and the protein relays that message. You need to memorize these 6 functions of membrane proteins. Luckily, there's a mnemonic device. So if you can remember JET-RAT, these are the 6 functions of membrane proteins. This is entirely stolen from a website called BioNinja. If you haven't visited BioNinja yet, you definitely should. It's pretty amazing. All right, so quick clarification on some of our proteins. So in this picture back here, there's this stuff stuck to the protein, kind of weird. We actually like to sometimes glue carbohydrates to proteins. Sometimes we also glue carbohydrates to lipids. And so glyco, kind of like glucose, is our prefix for sugars. Carbohydrates are sugars. So if I glue a little piece of carbohydrate or a sugar to a protein, I have a glycoprotein. This is an example of a glycoprotein. These little branchy guys, and you can see them here and here and here and here. These branchy guys are the carbohydrate branches that can get stuck to proteins, turning a protein into a glycoprotein. We also have glycolipids. So here we have a little piece of carbohydrate not stuck to a protein, but instead stuck to the phosphate head of a phospholipid, hence glycolipid. So if I stick a little piece of sugar to a lipid, glycolipid, stick a little piece of sugar to a protein, glycoprotein. And here we can see just rather a different kind of picture of those glycoproteins. So here's protein, here's the glyco part. Another protein, here's the glyco part of the glycoprotein. And then here is a little bit of carbohydrate, glyco stuck to phospholipid. So glycoprotein, glycoprotein, glycolipid. Good. All right. Some of our proteins in our membranes are what we call integral or transmembrane. These guys cross the whole entire membrane. They go from one side all the way to another side. An example of an integral protein is a channel protein. And these guys are literally just tubes that stuff can pass through. And what is so amazing about these tubes is that they are both polar and nonpolar. So we know that this part of the membrane is nonpolar. Nonpolar fatty acid tails of our phospholipid molecules, quite nonpolar. And so the outside of this channel protein, the outside of this tube, is also nonpolar, so that these guys can hang out together. The protein is not repelled by the lipid. Instead, it is attracted to the lipid. But the inside of the tube, where polar things are going to be passing through, is in fact polar. So we have a tube, and the outside of the tube is nonpolar, so it can stick in the membrane. The inside of the tube is opposite. It's polar, so that polar things like salt, can pass right through. So in addition to our integral or transmembrane, they go from one end of the membrane to the other. In addition to our integral proteins, we have peripheral proteins, and they just stick on the outside. So integral go all the way through. Peripheral proteins sit toward the outside. So here are some examples of integral proteins. You can see this is our phospholipid bilayer, 1 layer, 2 layers. This guy goes all the way through. This guy goes back and forth a couple times. This guy kind of spins around, but again, from one end to the other. These are examples of peripheral proteins. They're going to sit on just one side. This guy went halfway through, but not all the way through. So this is another example of peripheral protein. Another component of membranes that makes them remarkable is cholesterol. So cholesterol is this lipid molecule. It is another fatty molecule that is both kind of like our phospholipids, hydrophilic and hydrophobic. So this one little end that sits here by the phosphate groups of the phospholipids is polar or hydrophilic, as is this, right? The phosphate heads are polar. The little head of the cholesterol molecule is also polar. So they quite happily sit together. The rest of it is hydrophobic or nonpolar, and this sits within the nonpolar fatty acid tails of the phospholipid molecules. What is amazing about cholesterol is that it stabilizes the fluidity of the membrane. If the membrane gets quite cold, the cholesterol helps to keep it kind of mushy, lets it flow around, keeps it more liquidy. But here's the amazing thing about cholesterol. Should the membrane get too hot and start to melt, the cholesterol holds it back together. So depending on the conditions, the cholesterol is either going to reduce the fluidity if it's too hot or increase the fluidity if it gets too cold. So the purpose of cholesterol is to stabilize the fluidity, keep that fluidness just right. And these are the words that we just said. So we are both reducing and increasing fluidity. It's also going to reduce the permeability to small polar molecules. So the more cholesterol there is, the harder it is for things like water and ethanol to get through. It's going to prevent those tails from crystallizing. That's also going to help to anchor peripheral proteins. So the proteins that sit just on the outside, the cholesterol will help them kind of stick to the membrane because we don't want them to just float away. So let's practice just a little bit and then we'll call this lesson done. In the diagram, which structure is an intrinsic or integral protein. Remember, these are the ones that go all the way through. Find your answer in 5, 4, 3, 2, 1. Hopefully you picked a B. Because this guy goes from one side to the other side, it is our integral protein. Since we're looking at the picture, let's talk about the other letters. What is A pointing to? Well, that's the phosphate head of a phospholipid molecule. What is C pointing to? Looks like cholesterol to me. And then how about D? This little branch sitting on top of this protein? Well, that's going to be a carbohydrate, making this guy not just a protein, but a glycoprotein. Sometimes you might be asked to draw a diagram of a fluid mosaic model of membranes. Start with some phospholipid bilayers. Interestingly, we have one straight tail and then one bent tail. That also helps with the fluidity of the membrane. The straight guys like to stick together, but sometimes they stick together too much. The bent guys, they don't like to stick as well. They don't stack because they're kind of wrinkled. So having one straight allows the membrane to stick together. One kinked fatty acid tail, it doesn't get stuck together quite too much. You should add an integral protein and some more phospholipid molecules. We could add a peripheral protein and some more phospholipid molecules. We can add a glycoprotein. And hey, how about some more phospholipid molecules? And we can throw some cholesterol in there. I'm going to squeeze it in right there. Since it asks for a labeled diagram, we need to label. I'm going to call this a phosphate head. Fatty acid tails. Don't abbreviate when you do this for real, but you've been listening to me for a long time already, so I'm just going fast. But when you do this for real, don't abbreviate. Here we have a intrinsic or integral protein. Here we have a peripheral protein. Here we have a glycoprotein. Got our cholesterol. And then we can also always label the width of the membrane, just about 10 nanometers. That's a number you need to memorize, just about 10 nanometers. In IB, you get these little marks. You want to make sure that you have at least 5 things drawn and labeled in your diagram. We've got 1, 2, 3, 4, 5, 6, 7. Nothing wrong with having a couple of extras just in case. All right, that's all I've got for today. This is kind of a fun website game to play where you can build a membrane, put those pieces and parts together. I will link it in Canvas.